In this video, we're going to take a look at some of the structures that are found external to the cell wall of prokaryotic cells. So we'll start by talking a little bit about motility. That's just the ability of bacteria to move around. So motile bacteria are one that are capable of directed movement. And here we're not talking about Brownian motion. So there's Brownian motion. That's just the movement that occurs because water molecules and other dissolved stuff are kind of slamming into a prokaryotic cell. That is just a random kind of jiggling motion. With motility, we're talking about movement around that is directed, where the bacteria are moving toward or away from specific things. And the primary structure of motility is the flagella. It's a long, thin, helical appendage. So it is built of a protein called flagellin, and so it forms a helix, the flagellin does. And as it rotates, it pulls bacteria through whatever fluid that they are found in. It's anchored both at the cell membrane and at the cell wall by protein rings. And when ATP is supplied, then these two rings rotate against each other, producing rotational motion. They're arranged in a variety of ways. A bacterium could be monotrichous, having just one flagellum at one end of the cell. This is sometimes also referred to as a polar situation because there's a flagellum at one pole of the cell. So there is a monotrichous rod. They could be amphitrichous. Amphi means, think the same root as like ambidextrous, where you can use both your right and left hand. So an amphitrichous bacterium is one that has a flagellum at each end. So it's not polar, it's maybe bipolar? I don't know. Lophotrichous. Lopho is about having a cluster or a clump of flagella, and in this case, it could be at just one end or it could be at both ends. So here would be a lophotrichous bacterium. And if a bacterium is peritrichous, peri means around, so all over or all around the surface of the bacterium we find flagellum. So here is a peritrichous, again a rod. And then it's possible to be atrichous, having no bacteria, no flagella whatsoever. And this is the condition that we see most cocci found in, is that of being atrichous. So no flagella at all. Okay, so lots of different ways that we can arrange these flagella. So if they are all about motility, then how do flagella work? Well, if the flagella all rotate in a counterclockwise way, then, and now of course we're assuming that there are multiple flagella on the bacterium. So if all the flagella are rotating counterclockwise, then the flagella tend to bundle neatly together, and that creates very coordinated movement, or what is known as a run. So here we can see the flagella, they wind up being bundled together, and so this flagellum is going to, or this bacteria is going to move in the direction that the flagellum are spinning, and from that end, and it's going to be very directional movement. If, on the other hand, the flagella rotate in a clockwise direction, then the flagella all unbundle, and then that causes a tumble. And so we see, in response to this clockwise rotation, we see undirected movement, tumbling in all directions. So this then gives the bacterium the ability to track down specific cues. So it may be responding to chemicals or to light or to some other cue in the environment, but the bacteria will tumble until it catches a whiff of a chemical, let's say, and then it will run in the direction of that chemical. And if it starts to lose the chemical, then it stops and tumbles until it finds the chemical again, and then it will run in the direction of that chemical and tumble and run and tumble and run. And in this way, we see that there's movement. So here's a run followed by a tumble, another run followed by a tumble and a run and a tumble and a run and a tumble, and now a run again and a tumble and a run and so on. And that is how bacteria track down, for instance, a chemical that they are attracted to. So runs and tumbles being very disorganized and kind of flailing about, and a run being a very directional movement. So bacteria then, they exhibit what is known as taxis, or movement in orientation to some sort of stimulus. And if it's a chemical stimulus, then we talk about that as chemotaxis. If it's a light stimulus, it's referred to as phototaxis. And these taxis responses can be, or tactic responses, can be either toward or away from the source of the stimulus. If the bacteria is attracted to a chemical, then that is referred to as positive chemotaxis. If the bacteria is avoiding light, then we would refer to that as negative phototaxis. So negative is avoiding a chemical or light or some other stimulus, while positive tactical responses would be about being attracted to that chemical or that stimulus. Okay, so be sure to be able to distinguish between positive phototaxis and negative phototaxis, positive chemotaxis and negative chemotaxis. So the flagella is not the only structure that's found outside the cell wall. There may be structures that are referred to as axial filaments, or sometimes they're called endoflagella. So we see these in spirochetes. So there's a flagellum or set of flagella that lie between the cell wall and a structure known as the outer sheath. So this is still external to the cell wall. The outer sheath is an additional layer outside the cell wall, and arranged in kind of a spiral. So if this is the spirochete itself, remember spirochetes are the ones that are helical, then arranged inside this sheath, there's going to be spirals of these endoflagella that spiral around the bacterium. And you can kind of capture a sense of some of the spiraling here and the spiral filaments, the endoflagella of the spirochete. If we were to take a slice through it and look at it in cross-section, then you can see these axial filaments, the individual axial filaments here. And the reason they're all clustered on this side is because, recall, they will spiral around. And so if we were to do this slice a little further down the line, then the axial filaments would be arranged here. And if we went even further, like closer to your face, then the axial filaments would begin here. And so that gives this spiral configuration to the axial filaments. And so as the axial filaments, as they rotate, then that causes a corkscrewing motion of the spirochete through the substrate, through the medium that it's living in. Okay, so flagella and axial filaments are two external structures that allow motility. Pili are external structures that allow attachment to other surfaces. And so these are thin, hollow projections composed of the protein pilin. And there are two kinds of pili. There's conjugation pili and attachment pili. We're going to look first at conjugation pili, sometimes referred to as sex pili, because these are used, they're very long, and they're used to attach two cells to each other. So here we see two rods, and they are being held together by a conjugation pilus. So each of the bacteria reach out and they connect with their pili, and this it's understood to provide a pathway for the exchange of those plasmid DNA. So it's a way to move between two different bacteria some of the DNA that they have. And it produces then genetic variety among bacterial cells, as some cells get some types of DNA and other cells get others. So in a way, this process of conjugation is a little bit analogous to sex. There is no sexual reproduction in prokaryotes, but they do have the means to be able to share amongst themselves via conjugation the DNA that they have. Okay, so that is the conjugation pilus. And the other kind of pili that we see are attachment pili. These are short little hollow tube of pilin as well. They can also be called fimbriae, which is a weird term, but so fimbri, that's the first I, and then the AE also says E, so it's fimbriae. These are short hollow tubules that extend from the cell wall, and they can be used to attach one bacterium to another bacterium, or, and this is crazy, but they are capable, because of the surface tension of water, they can attach to the very top layer of the water. And so they can attach there at that air-liquid interface between the water and the air. Or they can use it to attach to a hard surface as well. And it's the attachment pili then that help to contribute to the pathogenicity, to the dangerousness of bacteria. If there are conjugation pili that will allow the bacteria to stick to the cells of other organisms, then those bacteria can be pathogenic on those poor host organisms. If they don't have those attachment pili, or if their attachment pili don't allow them to attach to an organism's cell, then it is not a pathogen to that organism. So it is their attachment pili that determines pathogenicity. And last of all, the last thing to say about attachment pili is that these attachment pili allow organisms, colonies of organisms, to form a pellicle. This is a layer of aerobic bacteria that are attached to each other and then to the surface of the nutrient broth that they're being grown in. And they use attachment pili to do that. And it's all about keeping these aerobic bacteria up near an oxygen source. And so we haven't talked about them yet, but aerobic bacteria are ones that require oxygen for their metabolic processes. Not every bacteria is aerobic. There are also anaerobic bacteria. Some of those anaerobic bacteria may even be killed by oxygen. And so if it's an aerobic bacteria and oxygen may be limited in the broth solution, then forming a pellicle is one way to keep the bacteria up where the oxygen can be found. So we're seeing here in test tube number 3, there's a pellicle that has formed at the broth surface. And so that's an example of that. So one bacteria would use its conjugation pili to attach there at the air-liquid interface. And then as it grows and divides, the bacteria continue to attach to each other and attach to more of that air-liquid interface, forming a pellicle. Okay, so the last thing to talk about that is external to the bacterial cell wall is a structure called the Glycocalyx. And it's a slippery, sticky outer surface on the exterior of the cell wall, and it's formed by polysaccharides. So we have these sticky, slippery polysaccharides that are found there outside the cell wall, forming the Glycocalyx. And there are two kinds of Glycocalyx that can be formed. One is a capsule, and this is a very thick protective layer. It's bound to the cell wall, so it's held there closely to the cell wall. And it's not just polysaccharides, but there are proteins in it and other materials as well. And it can be used, if it's thick enough, to cover up all of the proteins that are identifier proteins on the cell membrane and wall. Then that can help, the capsule can help the bacteria to hide from the immune cells of its host. So white blood cells won't be able to recognize it as being non-self. If it's a slime layer, this is much thinner, and it helps to trap nutrients and bind cells together as they're forming a biofilm on the surface of a material. And this thinner slime layer helps to protect the cell from drying out, and it can also help to protect the cell from antiseptic materials or antibacterial materials that might be used to attack this slime layer. And it makes it really difficult then to sterilize surfaces in hospitals, because as these biofilms form, it's hard, like a spray-on sterilizing solution may not be able to penetrate or get to the bacteria through this slime layer. Some examples of biofilms that are formed from the Glycocalyx plus all the bacterial cells that are hidden in the Glycocalyx are the plaque that we find on our teeth, or if you haven't cleaned the pet water bowl in a couple of days, then there'll be a slippery kind of layer on the inside, or maybe in the horse trough, or if you have horses, or anywhere that water stands for a long time in a vessel of some sort, these biofilm slimy layers will form where the bacteria have become thick on the surface. And so here we're seeing a biofilm. You can make out the cocci kind of embedded in this biofilm, and you can see how it kind of spreads beyond all the cocci and helps to bind the bacteria to the hard surface that it is resting on. Okay, so there you have it. Everything that you ever wanted to know about the external structures of bacterial cells, all the stuff that's external to the cell wall, including flagella and axial filaments, and the pili, both conjugation pili and attachment pili, and the glycocalyx that can be either exhibited as a capsule around an individual bacterium or as a slime layer that is shared amongst many bacteria in a biofilm. You know what to do if you have any questions.